Hello everyone, I'm Mitra. I'm a postdoc in the Evolution and Ecology Department at UC Davis. And today I will be sharing with you some of my work making use of gene expression dataset to look at how species would be responding to novel selective pressures. As the climate continues to warm, extreme events such as drought and fire continue to occur with higher intensity, and anthropogenic disturbances such as increased land use conversions continue to occur. A key question facing all of us is how will species respond to these multifaceted novel stresses, and in turn, how is it going to impact the biodiversity here on Earth? Specifically, as evolutionary biologists, as has been first demonstrated by the study by Clausen, Keck, and Heinze, we know that several populations of species are adapted to their current environmental conditions. So key question that I'm interested in is whether these adapted populations would be thrown off their fitness optima and experience maladaptation, and and whether there is going to be sufficient genetic diversity within populations and within species to help them respond to the rapidly changing climatic conditions and prevent rapid population decline. So in order to address this big question about how species respond to changing climatic condition, my talk today is going to address two aspects. First, I will be looking into can species respond to changing climatic conditions, and then move on to addressing the architecture involving this response. In order to do so, I will be focusing my work making use of this long-lived species of conifer, which is southwestern white pine. This species is predominantly distributed in the western parts of North America, as is highlighted by these purple polygons, and has this interesting disjunct distribution. For my talk today, I'm going to take a deep dive and focus only on the fragmented sky island populations, which is highlighted here in the northern part of the species range. The picture on the left-hand side shows you the sort of environments that these trees occur in. These trees are present in these high-rising mountain ecosystems, which are separated from each other by vast expanses of extremely dry and inhospitable desert ecosystems, which is not suitable for this cold weather-loving high-elevation conifers. And the reason here I've chosen to focus on the northern part of the species range is because as anthropogenic disturbances increase, several species or populations of species will experience high levels of population fragmentation. So this essentially provides us with a very useful setup to evaluate how this sort of population fragmentation might impact the ability of species to respond to novel multifaceted selective pressures. Okay, so in order to get at the novel aspect of climate change, I made use of a space for time substitution design. And what this essentially is an approach whereby I used several individual trees that originated from 10 different populations represented by the green triangles, and evaluated their performance at two sites, which is Bear Spring and White Pocket shown here in orange, by growing them at these two individual sites. And despite some of the differences between these sites, the key aspect that helps us evaluate their response to changing climatic condition is that both of these sites expose the trees to really dry conditions, which we expect that several species in the western parts of North America would be experiencing under all future projections of changing climatic conditions. The figure in the bottom is simply highlighting is simply showing you a snapshot of the extent of drought stress that these trees are exposed to at both of these common garden sites. The one on the right-hand side was taken during the sampling here, and you can see that several of the trees that were grown in this area were not able to withstand the high intensity of drought stress that they were exposed to. So typically in these sort of common garden experiments or space for time substitution designs, we make use of organismal level traits in order to evaluate the adaptive potential of individual traits. However, this is often based on a priori information about what might be adaptive, but specifically with regard to changing climatic conditions and novel selective pressures, we don't always know what sort of traits might be of importance to help species respond to novel stresses that they may encounter. So in order to avoid this ascertainment bias, I decided to make use of a gene expression dataset or transcriptomic profiles as a quantitative trait in order to evaluate the adaptive potential of species. So making use of all of the individual seedlings that were represented in the common garden, I obtained a total of 27,000 gene expression traits, which were treated as individual quantitative traits. For each of these, I fit a mixed effect linear model, which took into consideration aspects of the garden, as well as the mixed set design that was used in order to estimate the genetic component underlying the noted trait variation. From this mixed effect model, I pulled out variance component and estimated a metric called as QST, which varies from 0 to 1 and represents the extent of among population trait differentiation. What we are really interested in here is identifying traits which have high values of QST, because what it would be indicating is that the populations that were represented in these novel environments experienced different selective pressures or different environmental conditions at the native site, which has impacted their ability to respond to these novel climatic conditions in different ways. And this would mean that these traits that exhibit variation are likely adaptive and represent some amount of genetic diversity, which could be useful for these species to respond to the future climatic conditions. However, high values of QST can also result from neutral processes, such as the extent of gene flow amongst the sampled populations. So in order to account for this, I made use of an independent data set through which I estimated a very similar measure, which is called as FST, and it also ranges from 0 to 1. And so now for each of these gene expression traits, I can compare their QST values to FST, making use of a formal statistical test to identify if there are certain traits that exhibit adaptive potential, which is essentially addressing the first part of whether species can respond to changing climatic conditions. So in this plot, the y-axis is representing the QST values. And what we are really interested in is identifying traits which fall above this black dotted line, which is giving us the 95th quantile of FST, which is the null empirical pattern of population differentiation as a result of differences in the extent of gene flow amongst the assayed populations. So on the left-hand side, I will be highlighting traits which are adaptive in bare string as they fall above this black dotted line. And on the right-hand side, I will be highlighting traits which are adaptive in white pocket. And so overall, across the 27,000 gene expression traits, we find several that exhibited really strong patterns of adaptive trait differentiation, indicating that the differences in environmental condition where these populations originated from likely caused these individual seedlings to respond differently to the novel climatic conditions that they were exposed to. So here I'm only representing about 1% of gene expression traits out of the 27,000 that had really high values of QST after accounting for the null expectation shown here by the black dotted line. So for the first part, what I've shown you is really strong signals of adaptive evolution in these three species to novel climatic condition, highlighting that there is in fact some potential of these trees to respond to the changing climatic conditions. With that, I can now move on to addressing the second part, which is thinking about the architecture that facilitates this adaptive response in forest trees. When we think about the architecture, there are a bunch of different aspects that go into it. But for the purpose of today's talk, I will be focusing on two important aspects. The first is highlighted in the slide, which is basically identifying the genomic regions that underlie trait variation, which could be aspects such as differences in relative growth or the extent of drought stress that individual seedlings and the populations that they represent are able to withstand. And so what here we essentially do is look at various regions across the genome and identify not only the genomic regions associated with this trait variation, but also the extent to which they are associated with the variation underlying this trait. The second aspect about genetic architecture that I'm really interested in is the pattern of covariance. And this is key because traits do not exist in isolation, and the way in which they covary with each other and can impact the evolutionary potential of species. For instance, if the axis of selection is parallel to the pattern of covariance between the traits, then we expect the traits to have a significant contribution towards evolutionary change. However, if the axis of selection is orthogonal or opposite of the pattern of covariance between the traits, then we expect that the traits may not contribute that much towards adaptive evolution or evolutionary change, and in worst case scenario might even impede evolutionary change. So we can do this in our study design by taking these 27,000 gene expression traits and turning them into a biological network, wherein the circles are representing the traits and the lines connecting them are representing the covariance between each of these traits. So we can also color code the sort of biological network to highlight the extent of constraint that is experienced by each trait as a result of where it is situated in the biological network and the amount of information or covariance an individual trait experiences. In general, we expect traits which are located towards the periphery of this biological network to have fewer connections and less constraint, and as a result, evolution is more likely to sort of tinker with these peripheral traits in order to help individual trees fine-tune their response to changing climatic conditions. So the adaptive traits that we identified in the first part, we expect them to be situated more towards the periphery. We can test this expectation by making use of the adaptive traits from the first part and evaluating using a statistical framework whether there are certain parts of the biological network that they seem to be more so enriched in than in others. So we did this for both of the gardens, and what we find is quite opposite of the expectation that we started out with. In this wall plot, the circles or the dots are representing the gene expression traits, and what we notice is that most of the traits that exhibited high differentiation are located towards the center of this biological network. The figure on the right-hand side is giving you a simplistic representation of our result, and what this shows us is that adaptive traits are located at the core, despite the core traits likely experiencing high constraints in general. And this is because if you are to play with these core traits, you would be changing a bunch of different aspects about how a specific individual functions. And so I set out today in this talk to ask an important question about can and how the species be responding to changing climatic conditions. And the key takeaway or conclusions from this two-part evaluation is that gene expression profiling, when coupled with a space for time substitution design, is a really effective way to attain a more holistic view of the future adaptive landscape of many species of many natural species of plants. Specifically in the context of long-lived species of trees and also systems that experience high levels of population fragmentation, what we notice is that despite the high levels of fragmentation and despite experiencing normal climatic condition, these trees exhibited some really strong potential to respond adaptively to future climatic conditions. And we think this is likely a result of most species of forest trees having high levels of genetic diversity and having high effective population size, which likely is a key contributor in helping them respond to novel selective pressures, even if it happens really rapidly. In the second part of my talk, I find some pretty interesting results. And what I essentially show here is that when the fitness optima is shifted rapidly, populations will likely undergo a shift in their genetic architecture, causing traits which have high patterns of covariances and are likely pleiotropic to be more involved in aiding an adaptive response. This result at first seems really counterintuitive because it seems risky to change a bunch of different traits that co-vary with these core traits, and because it will likely influence how a specific individual functions, which you may think could actually be deleterious in general. However, both theoretical as well as empirical studies have demonstrated the prevalence of this sort of architecture being beneficial, at least in the initial stages when the populations are really far away from the present fitness optima, as we may expect when the climate shifts rapidly and climate shifts more rapidly than the generation time or the turnover that we may expect for certain species. And with that, I'd like to acknowledge my collaborators and my funding sources. And please get in touch with me and look up our paper on bioRxiv if you are interested further in getting into the details of the work that I've presented today. And